Hello and welcome back. In this lecture we're going to tackle the very interesting system called the lymphatic system and then the functional division of the lymphatic system, which is called immunity. So I don't know how you think about the human body, but I think of it as like a salad bar. You know, when you go to the salad bar, there's all kinds of things that you can eat that's available for you on the bar. And the human body is the same way for many different kinds of organisms. So for example, we have all different kinds of creatures that want to come in and eat our bodies for food. And so thankfully we have a whole system that protects us from being eaten. So we have things like, let me go back here. So we have things like bacteria that want to eat us. And so this would be an example of a bacterium right here. This is Helicobacter pylori, which is the bacteria that will get into your stomach and eat the inner lining of your stomach. So there's all kinds of forms of bacteria that want to eat us like they're eating at a salad bar. You know, there's nothing in the human body that the bacteria can't eat. You know, we're made of proteins and carbohydrates and lipids and nucleic acids. Those are all food for all these different living organisms. Now, not all bacteria are bad. We have to have some bacteria in our intestines and on the surface of our skin that are mutualistic bacteria that benefit us, but there are some 100 forms of bacteria that do cause humans disease. Also trying to enter into the human body are these things called viruses. This happens to be the Ebola virus, and this one comes in and will kill the cells lining your blood vessels and cause bleeding to occur all throughout the body. So viruses don't really eat a human being, but they do infect our cells, and in the process of infecting our cells, they cause the cells to die and new viruses to be released. There's a whole host of different kinds of fungi that like to enter into the human body and eat it. This one happens to be athlete's foot. So fungi will come in and eat at the salad bar. And then there's also protistal organisms. So we've talked about bacterial organisms, viral organisms, fungal organisms. Then there's a whole host of protistal organisms. This happens to be trypanosoma, which causes African sleeping sickness. But there's also malaria and giardia and a whole host of other protistal organisms that love to come in and feed on the human body. We also have in the world some really interesting parasites. We have different kinds of roundworms. This happens to be hookworm, and this one is ascarid worms. These are very common, infecting billions of people. There's also some flatworm parasites like tapeworms and flukes that also want to come in and feed at your salad bar, your body. And this particular fluke right here has a blood meal inside of its intestines for you to enjoy. Well, there's many other kinds of parasites. We have things like chiggers in our area, and these cause little red whelps to form on the human body, but typically they're not deadly. We also have a whole host of fleas that can carry disease, but in and of themselves they do feed on your body. We have body louse. These are the creatures that feed on skin cells, and they, just recently there's been an article just been published that thinks that suggests that lice were what caused the spread of plague in medieval Europe instead of fleas. We also have ticks, and we have various things like mosquitoes. So all of these organisms come to the human salad bar to feed. Now, we're not just a plain salad bar. This salad bar right here doesn't have an immune system. You just go in and feed. But luckily, our body has an immune system in order to destroy these organisms. And so what the next couple of figures are going to show is our lymphatic system. These are all the parts that are going to aid in allowing us to have immunity. So on the left-hand side of this particular graphic, we have lymph nodes and lymphatic vessels. So besides arteries and veins and capillaries, we have a whole other set of vessels called lymphatic vessels that drain and filter all the fluids that are inside of our body. And you can see how they're distributed through the body, so they are everywhere inside the body. And so the lymph vessels carry the lymph fluid, and the lymph nodes are going to be the filters of that fluid. They're going to have large concentrations of phagocytes lymphocytes that are going to aid in causing immunity to occur. And hopefully those viruses, bacteria, cancer cells, and other creatures that come in will be destroyed by those cells there. We have a whole lot of other lymphoid tissue and lymph organs that are going to aid in this process of immunity. So we have tonsils, we have the thymus, the spleen, we have mucosa-associated lymphatic tissue, also known as MALT. This tissue is found in the digestive, respiratory, urinary, and reproductive tracts, and they're right underneath of the mucosa, and they are high concentrations of phagocytes and lymphocytes, which are going to aid in immunity. Just looking at the lower part of the body, we do have the appendix as another lymphoid tissue, and we also have red bone marrow, which is going to be the original source of all the lymphocytes and phagocytes that will aid us in immunity. Okay, and as you can see, we have lymph nodes and lymphatic vessels all spread throughout the body. All right, well, let's talk about those lymph nodes and lymphatic vessels just for a minute so we can understand how important those things are. So you see here in red arteries, we see also here in blue veins, and then we see the capillary beds. But in addition to arteries, veins, and capillaries, we have this whole other set of fluid conducting tissue. And this particular green, these vessels here are called lymphatic vessels. And what they do is they will absorb or reabsorb interstitial fluid, and that fluid will flow through these lymphatic vessels. Now, there's no blood cells inside of lymphatic vessels. It's merely the fluid, the tissue fluid. So all of this fluid that's in your tissues will eventually flow into the lymphatic vessels, and we'll call that lymph flow. And just looking a little bit more specifically here, you can see in cross-section here a little capillary. And you can see that the capillary doesn't allow blood cells to leak out, but it does allow the fluid to leak out, and it becomes interstitial fluid. Well, those fluids are going to, bathe the tissues. They're going to oxygenate the tissues. They're going to nourish the tissues from the materials coming out of the bloodstream. And then eventually they'll enter into the lymphatic system. Perhaps they'll be reabsorbed back into the bloodstream. But they'll enter the lymphatic system. And inside the lymphatic system, if there's any bacteria or viruses that are in or cancer cells or other kinds of organisms that are in the interstitial fluid, if they flow into the lymphatic vessels, they're not going to find a very hospitable place, because we're going to have all kinds of cells there that are going to aid in destroying those particular parasites or microbes that are deadly to the human body. So all the lymph will eventually flow. So they'll go through these afferent vessels. So they'll come in through the lymphatic vessels, through these afferent vessels into the lymph node. And in the lymph node, the little any kind of foreign agent is going to enter into the lymph node, and it's going to have all these phagocytes and lymphocytes that it has to go through. And what is going to happen is, in the lymph node, it's going to be like a filter, so that all those little critters that come into your body get into the lymph. They're going to go into lymph nodes and be destroyed by all of the millions and millions and millions of phagocytic cells and B cells and T cells or lymphocytes that are going to be in there. So once the fluid is kind of like, in quotation marks, cleansed, it's going to leave the efferent vessel to flow into other lymph nodes and eventually back into venous blood supply. Now, we do have valves in these lymph nodes to ensure that the flow of lymph is going to be in a one-way flow. And so that's a little bit about lymph nodes. Now, when you go to the doctor and you have like a sore throat or some kind of infection, they're going to feel all over your body, and what they're trying to feel for are swollen lymph nodes. A swollen lymph node is a sign that you are fighting something off. So that's a good sign that you have an infection. Well, this is just showing you have arterial blood, you have venous blood, you have capillary bed. So when the capillaries allow fluid to leak out, that becomes interstitial fluid. Some of the interstitial fluid will enter into the lymphatic capillaries. This will go into lymph flow, which will enter into lymph nodes to be cleansed. Then those lymphatic vessels will eventually will eventually take all of the lymph and add it back into venous circulation. So, the lymph vessels are very well known, and we have several of them that are pretty key. I'll only ask you to remember a couple of them. The right lymphatic duct is where you have a vessel that drops blood off into the right subclavian vein. So you can see the right subclavian vein right here. So that material is dumped off into venous circulation. So all that fluid goes back to your venous circulation, becomes blood plasma, and eventually it'll leak back out when it gets to a capillary bed. We also have the thoracic duct. Okay, it's located right here, and that's going to drop off lymph into the left subclavian vein, and then it goes into venous circulation and eventually becomes part of the plasma. Now you can see the drainage of the right lymphatic duct drains this portion of your body. So all the lymphatic vessels in this portion of your body will be put into that right lymphatic duct and then drop back into venous circulation. The rest of the body eventually all collects and drains to the thoracic duct, which then drops it off into venous circulation. So you can see a larger portion of your body is serviced by the thoracic duct, and that's where the lymph drops back into venous circulation. All right, let's talk about a few of those parts that are part of the lymphatic system. So I'm sure you've heard of tonsils before. Tonsils are going to be found in various places in your behind your tongue, in your pharynx, and at the back of your throat. So we have the palatine tonsils, we have the lingual tonsils, and then we have the adenoids or the pharyngeal tonsils. They're also known as adenoids. And what a tonsil looks like when you look at it, this is what it looks like under the microscope. And so this is where it's attached to the wall of the throat or whatever part it's attached to. But you can see all of this purple that you see here are nuclei of lymphocytes that have been stained. Okay, so there's tons and tons and tons of lymphocytes and other lymphoid cells that are in there that are fighting infection. And there are crypts. So that's the entrance to a crypt. And you can see this crypt goes deep inside of this little nodule of lymphatic tissue that we call a tonsil. Okay, so those are little crypts. So the tonsil is kind of an interesting organ. What it does is it basically, when you eat food or when you breathe air, it allows for little microbes to enter into those crypts, and then your body learns it's a place where your body learns to recognize all the local pathogenic organisms. Now, sometimes it can be overwhelmed by pathogenic organisms and you can get an infection in these things. Some people are sensitive to having swollen tonsils all the time. They're always infected. And then we have to do a tonsillectomy where we take and remove those tonsils, or we'll remove the adenoids if those become swollen or inflamed. That can be kind of dangerous when they get swollen and inflamed. So let me erase all the ink. So if your adenoids swell and you can actually have problems bringing air into your respiratory system. These can get swollen too and cause for painful swallowing. If they keep on getting infected, they'll remove them to reduce the infections coming into your body. Related to the old tonsils are tonsil stones. A lot of people get tonsil stones, and I just thought I'd kind of share with you what those are. So remember that your tonsils have these crypts. Okay, so these are like little channels where the bacteria comes in, but there's also fluids and food and things that can get into those tonsils as well. Well, if you have really deep crypts and really big crypts, sometimes you get these things called tonsil stones, which is where you have a collection of, bacteria, dead bacteria, food that you've eaten, and things like that, and they'll actually kind of harden as they accumulate, and they'll actually fill up the little crypts of your tonsils. You can take a something like a Q-tip and press on the tonsil, and those stones will pop out. But there seems to be some people that are more susceptible than others. I personally have never had a tonsil stone, but I know many people that have. So they oftentimes are very foul smelling because they're full of bacteria and decaying food and beverage. Something related to the throat that is something important for you to understand is strep throat. Okay, strep throat is where you have a bacterial infection that's in the back of your throat. And it's important that you understand the symptoms of strep throat. There can be viral, sore throats, and you can have strep throat, which is from a bacterium. So some of the warning signs of strep throat include sudden and severe sore throat along with painful swallowing. So pain is definitely part of strep throat. A fever of 101 degrees Fahrenheit or higher. Red and swollen tonsils with white patches. You can see over here in the graphic what those little white patches look like. And what that is basically pus that's forming from bacteria being killed and white blood cells being destroyed. Skin rashes and body aches, headache, nausea, vomiting, swollen and tender lymph nodes. Okay, so if you don't have all of these particular symptoms, then it may be a viral infection. So strep is a pretty serious infection. There is a strep test that they can do at the doctor's office. And if you have bacterial strep, then antibiotics are warranted for taking. Antibiotics, of course, don't work against viral infections of the throat. So it is important to get, medical help when you have a really painful sore throat, and especially with the fever and the swollen lymph nodes. So this just shows you the throat redness, white spots, swollen tonsils, so you can kind of compare and contrast normal versus abnormal. So mucosa-associated lymphoid tissue, or MULT tissue, this is going to be found in your respiratory passages, reproductive passages, and around digestive organs. And what these places are just aggregations of lymphocytes and phagocytes, so that if anything penetrates the wall of the mucosa, so the mucosa being this epithelium, if it comes into this lymph node, this MULT tissue, it'll be destroyed by that tissue. So it's really good that you have that tissue to prevent infections from getting deeper into the body. Another lymphatic organ will be the thymus. It produces hormones like thymosins, which stimulate the production of lymphocytes. The thymus is also a place where lymphocytes go. Lymphocytes like T lymphocytes are going to go in order to be educated about local organisms, and also to teach them about the proteins that are on the surface of your own body cells. You want to teach T cells not to recognize your body and respond to your body, because that would cause a destruction of your body's tissues. We'll talk more about the thymus in a little bit. Another associated lymphatic organ will be the spleen. It's like a super huge lymph node. It's a filter of blood, and it helps you to destroy microorganisms that enter into the body. So the spleen's located, as you can see in the graphic over here, and this is just a transverse section through showing you where the spleen's located. And our last tissue I'll talk about right now will be the bone marrow. The bone marrow is your source of all of your phagocytes and lymphoid cells. So that hemocytoblast is the mother of all stem cells in bone marrow, and it has the ability to become all the different kinds of phagocytes and lymphocytes that your body produces. Okay, now let's talk about so that's kind of like the anatomical division of the lymphatic system. Now let's talk about immunity, which is the functional aspects of the lymphatic system. So this is how your body fights infection off. So we're going to talk about innate immunity. Innate immunity is a rapid response to a broad range of microbes, and it's something that you're born with. Acquired immunity is a slower response to very specific microbes, and your body has to be taught how to do acquired immunity. So we're going to basically talk about in this lecture innate immunity. We're going to begin by talking about all kinds of external and internal defenses against invading pathogens. So in this part, I want to talk about physical barriers. I want to talk about all the wide range of phagocytes that we have to destroy invading organisms. I want to talk about how we surveil and defend ourselves. I want to talk about chemicals that we produce called interferons, which are messengers that coordinate various kinds of defenses against infections. I want to talk about some disease-fighting proteins like complement. I want to talk about the role of inflammation and fever. All of these will be innate defenses that we have that you're born with. As soon as you come out of the womb, you have these defenses working for you. Okay, so we'll talk about innate immunity. So really our first line of defense is surface membrane barriers. So our skin is one of the great physical barriers. It resists acids from bacteria, it resists bases, bacterial enzymes. It's almost indigestible to bacteria. We have created from sebaceous glands. We have sebum that basically is liberated on a hair follicle. It spreads over your skin and it's toxic to many forms of bacteria. You know, sweat inhibits microbial growth because it's a salty solution with ammonia and urea in it, and it's not very hospitable to microorganisms. Keratin is a protein that is made by keratinocytes that make up your outer layer of skin, and those are resistant to that protein's resistant to bacterial enzymes. Cerumen is earwax, and it traps microbes and repels insects. So we have a wide range of chemicals and solutions that we secrete on our skin that help to resist microbial growth. We also have, in addition to skin, we have mucous membranes. Mucous membranes line all of your mouth and your respiratory passages all the way from into the lungs and to the anus. You can go all the way from the mouth to the anus and still be in a mucous membrane. One of the great things that mucous membranes produce is mucus. Mucus is a mucopolysaccharide that's made of protein and sugars, and it's sticky and it traps little microorganisms in it, thus helping to prevent you from getting those organisms. Nasal hairs are there to protect you from microbes coming in and from larger parasites coming in. They are sensory and they're also have a filtering nature to them. If organisms do enter in through the mouth, they'll go down to the gastric juice. There are acids and all kinds of enzymes, proteases that will digest organisms that come in. When you have mucus coming up from the trachea, you know, you oftentimes all day long swallow that and it goes to the stomach to be killed in gastric. Anything that's microbial will go down to the gastric juice and be destroyed. We also have that, that mucosa associated lymphoid tissue lining all of the mucous membranes just about in the human body. Saliva is going to contain lysozyme, and it's going to help neutralize some bacteria. Tears also contain an enzyme called lysozyme, which can kill components of bacteria. Urine is acidic, which every time you urinate, it washes the little tubes, the urethra, and since it's in the ureters and the urinary bladder, it's going to be acidic, helping to wash bacteria and fungi that might be coming into your body with acid. So the vaginal canal is also acidic. It produces lactic acid, and that helps to inhibit microbial growth, such as yeast and bacteria. And then cilia are also associated with mucous membranes, and they will help to sweep the mucus along and any microbes that might be trapped inside of that mucus. So our second line of defense are the phagocytes. We have many different kinds of phagocytes. Probably the one that's most common in your bloodstream and throughout the body are the neutrophils. This is a very, very, very abundant white blood cell. We also have monocytes inside your bloodstream, and when they come out of the bloodstream and enter your tissues, they become macrophages. These are really large bacterial-eating phagocytic cells. In our bloodstream and leaking out of our bloodstream are eosinophils, and these protect against parasitic worms. So, in our blood, neutrophils make up about 52 to 62% of all the blood cells, white blood cells, and they have an interesting characteristic. They have a lobed nucleus, and their cytoplasmic granules stain very light purple. The cytoplasmic granules are, the place that contains the digestive enzymes. When they engulf bacteria, they're going to use those antimicrobial enzymes in those lysosomes to kill microbes as they come in. If you do have an infection or oftentimes hemorrhage or major bleeding, you will have neutrophilia, which is when they do a CBC, you'll see, which is a complete blood count, you'll see a whole large number of neutrophils when you do a blood smear and look at it under a microscope. So monocytes make up a smaller percentage of the circulating white blood cells. They have that kidney-shaped or oval-shaped or horseshoe-shaped nucleus. They engulf internal parasites and cellular debris. You oftentimes will see monocytosis when you have certain kinds of infections like brucellosis, tuberculosis, rickettsia. These are bacterial infections. And you'll see monocytes in huge numbers when you have those types of infections. And this is just a monocyte gobbling down. It appears to be like a yeast cell. So when these phagocytes work, they basically will take the microbes. You can see up here, the microbes are coming in. There will be little extensions of the white blood cell called pseudopodia that will wrap around the microbes. And you can see the little sequence here. So as those pseudopodia reach out and wrap around, it'll then pinch off into a vacuole. So this is called a phagosome. Okay, so this little vacuole is made in the process of phagocytosis. So we do have lysosomes, which contain enzymes. These things will link together. The phagosome and the lysosome will link together, forming a phagolysosome. And so the little chemicals will go over into the vacuole part, digest the microbes, and those microbes will be spit out because the creature's been destroyed. So, neutrophils and macrophages, they kill microbes in many different kinds of ways. So one of those is, like I just showed you, the lysosomes will join with the vacuoles containing the microbes and digest them. So these cells also have the ability to make hydrogen peroxide, which is very toxic to these organisms. So that's called an oxygen-dependent killing mechanism. You can also, not you, but the neutrophils and macrophages can generate these really dangerous halide ions, which are charged atoms, and these anions. And so they form iodine, chlorine, and bromine. These things are very, very super damaging to microorganisms. So the vacuoles that these microbes are contained in can actually be caused to lower in pH. Lactic acid can be pumped in, and hydrogen ions can be pumped in to lower the pH of those vacuoles, and thus help to inhibit microbial growth or even damage them. So there's all kinds of enzymes, lysozyme, elastase. These things will digest cell walls, thus killing microorganisms. And then there's also chemicals like lactoferrin, which bind up iron and inhibit microbial growth. There's many other things. These are just a few things that the neutrophils and macrophages can use in their arsenal to kill microorganisms. Eosinophils are very rare in blood. They do have cytoplasmic granules that stain red, and they are very likely to kill parasitic worms. What they do to a parasitic worm is they'll lock onto a parasite and then dump their lysosomes onto the parasite, helping to digest or irritate the parasitic worm. So you see eosinophilia, so a high level of eosinophils in a blood smear. during heavy worm infestations. So I just want to show you how these cells can actually leave, how these neutrophils, monocytes, and eosinophils can actually leave a capillary and move to a site of infection. So here we have a needle or a pen that's been stuck into the skin. You can see it has microbes all over the surface of it. Those little microbes are green, and when you have tissue damage, the little signals will be given off that will diffuse down to the capillary. The capillary, you can see, swells. It becomes dilated, and it becomes more permeable. So so we'll actually have these white blood cells will actually migrate out of the capillary, and they'll move towards the site of where these chemicals are being liberated from, and then they'll begin to eat the microorganisms. Eventually the blood vessel will constrict and go back to its normal resting diameter, and the cells won't be crawling out. This process is called Daya PD SIS. It's the way that the cells go to, the way the white blood cells go to infection. And they follow, you almost think of, they sniff their way using these little chemical trails. They sniff their way, I know they don't have noses, but they sniff their way to the site of infection and help to digest all those microbes and rid the body of those microbes. So as the cells are circulating around, there are chemical clues that are basically given from tissues, as you can see right here. Those chemical cues will actually cause there to be receptors on the surface of the blood vessels. These circulating white blood cells will attach to these little receptors, as you can see here, and then they'll squeeze their way in between the simple squamous cells making up the capillary, and then they'll migrate to the source of where those chemicals are coming from. So it's very complicated and very specific. You actually have receptors on the surface, and these cells have to receive that message, and then they have to squeeze through the squamous cells, and then they have to travel to the site where those chemicals are coming from. Well, another line of defense we have is immune surveillance by natural killer cells. Natural killer cells, also known as NK cells, attack cancer cells, and they also attack virally infected cells. This is pretty important. If a virus is inside the cell, it's kind of maybe hidden from the immune system, kind of. But natural killer cells do have the ability to find those virally infected cells and destroy those cells. This is what the natural killer cell looks like in a blood smear, and this is how they work. So in our first step of natural killer cells working, they basically will recognize and adhere to an abnormal cell. So here we have the abnormal cell in yellow, natural killer cells over here in blue. Now this cell that's been infected or abnormal will present various kinds of proteins on its surface that are recognized by the natural killer cell. If the natural killer cell attaches to that cell, that abnormal cell, and it recognizes that antigen, it will realign its Golgi apparatus, which is going to be the part of the cell that's going to take proteins made from the ER and package those proteins into little vacuoles that will then be exported and dumped onto the cell that it's stuck to. So here we have the secretion of this protein called perforin. So the Golgi apparatus is making a huge number of these vacuoles. They're being exported onto the surface of the abnormal cell. These perforins, these perforins make holes in the target cell, and it causes the cell to basically be destroyed, blow up, or water flow in, and it destroys the cell. And then you have other, you have macrophages and other cells that will come up and gobble up the debris. So these are very, very dangerous little pores that form and destroy the abnormal cell. So this antigen could be something like on a cancer cell. So cancer cells will make abnormal proteins that natural killer cells can recognize. These antigens can also be viruses that are inside of a cell that the cell is presenting to the natural killer cell and showing that, hey, this thing is inside of me, and so the cell will be killed by the natural killer cell. Okay, so another line of defense we have that we're innately born with are interferons. Interferons are small proteins that are released by activated lymphocytes, macrophages, and tissues that are infected with viruses. You can think of these things as being like kind of like local hormones. And so we have interferon alpha. This is produced by cells infected with viruses. So if let's say this cell right here is infected with a virus, it will begin to release this alpha interferon, and it attracts and stimulates natural killer cells to come and kill it. It also will enhance its own resistance to viral infection. So it'll produce proteins that will help to be antiviral proteins. So we have interferon beta. This is secreted by fibroblasts in connective tissue, and it slows inflammation in damaged areas. So it modulates inflammation, which can be very damaging. Inflammation can be very damaging. We also have interferon gamma. This is secreted by T cells and natural killer cells, and it stimulates macrophages to basically, do their thing, which is to eat up microorganisms. Okay, so we have a whole host of these little proteins called interferons that will aid in various immune reactions. Complement's kind of an interesting aspect of innate immunity. There are all kinds of these proteins that are made by the liver and put into bloodstream. There are around 30 of them that we've discovered so far, and these proteins work together to form what we call the complement system. It's a whole system that's organized to destroy microorganisms and to enhance or complement activities of phagocytes and these disease-fighting proteins called antibodies that we'll talk about a little bit later. They're very sophisticated, and they're usually made in an inactive form, and then they activate themselves once you have an infection. So let's talk about the classical pathway first. This happens to be the most rapid and effective activation of complement, and it occurs through antibodies stuck to the wall of bacterial cells. So when these antibodies bind to the bacterial cell, they're going to attach to complement protein C1. C1 has to be attached to two antibodies for it to become activated. So if antibodies aren't stuck to bacteria, then C1 won't become activated in this classical pathway. Well, when C1 is attached to those antibodies, it's going to act as an enzyme, and it's going to take other complement proteins, and it's going to basically cut them and then cause them to become activated. So there are complement proteins that are circulating around in an inactive form, and they have to be basically activated in order for them to work. So in this particular example, we have C3 is basically splitting into two more complement proteins called C3a and C3b, and these will activate various kinds of inflammatory responses. So C3a is responsible for going through and activating inflammatory responses. CB3 will attach to the bacterium, and it's going to cause phagocytosis to occur, as you'll see in just a minute. Each of these other pathways also uses CB3, so I'll talk about how CB3 works in just a minute or so. Okay, so that's generally how the classical pathway works. Let me just remind you. We have to have antibodies stuck to the bacterial cell wall. The complement C1 will attach to those antibodies and become an enzyme. This enzyme will cleave various complement proteins. Typically, it splits C3 into C3a component, C3b component. C3a is going to activate the immune system. CB3 is going to cause phagocytosis of that microorganism. So the lectin pathway is a little bit different. So in the lectin pathway, you have a protein mannose binding lectin protein that will actually join with the surface of bacteria. And once this MBL forms a complex, it's going to split C3 into complement C3 into C3A and C3B. This is going to activate the immune system. This is going to stick to the microbe and will enhance the phagocytosis of that microorganism. So this is a little bit different. You know, we're going to be binding to various components on the bacterial membrane with a very specific protein that we create. That's going to basically split C3 into its activated components. In the alternate pathway, this is going to basically this one's going to interact in a slightly different way. So the alternate pathway begins with several complement proteins. So it begins with this protein called properidin, and properidin is going to interact in the plasma, causing C3 to split into C3A and C3B. Okay, so this particular protein, properidin, is going to sense that you have bacteria or parasites or virally infected cells inside your system, and it will activate C3, which will split into C3A and C3B. This will activate the immune system. This one's going to stick to those organisms and cause perhaps phagocytosis to occur. Okay, so these are different pathways in order to utilize the complement proteins. Now, C3b, once it's stuck to a bacteria, it's going to activate some other complement proteins that are in the plasma of your blood, and it's going to cause them to form a membrane attack complex. So these various components there will be urged on by C3b to cause all these pores to form on the surface. Here's what it looks like under electron microscopy, but it causes all these pores to form on the surface of the bacteria, and it will cause inrushing fluids to burst and to cause lysis of those microorganisms. Okay, so all those pores that will form from the membrane attack complex will cause the bacteria to be destroyed. So not only will CB3 work in that way, but C3b will also target cells that the molecule is attached to for phagocytosis. And it does it through the process of opsonization. Opsonization is where the CB3 will be a molecule that will signal phagocytes to eat that particular cell. C3b, let me get this right. So C3a, that little molecule of C3a, is going to cause mast cells to release histamine, and it will actually activate various components of the immune system to become stimulated. Okay, so that's how C3a is going to have its work. So it will cause inflammation, activate phagocytes, accelerate the flow of blood to that region, thus enhancing the immune response. So complement's really important in immunity. So there's also inflammation is another thing we're born with, and there are four characteristics of inflammation. I'm sure you've experienced this. Redness, pain, heat, and swelling are all components of inflammation. And there's three stages to inflammation. First stage is vasodilation. That is, your blood vessels, the diameter of your blood vessels, your little capillaries will increase in size and increase in permeability. So you'll have more blood flowing to that area where inflammation is occurring. This is caused because of the release of histamine, various prostaglandins, and leukotrienes. These are molecules released by cells like mast cells. Once they sense there's something wrong, they release these chemicals and vasodilation will occur. So this causes some of the redness and the heat and the swelling that you see characteristic with inflammation. Inflammation, you also have a stage, the movement of phagocytes to the site of injury. And then you have, eventually, after the phagocytes eat all the microorganisms, then you'll have tissue repair. So there are stages to inflammation, 3 stages of inflammation, and this is a really important characteristic of immunity. Not only will those things occur, but the area that's infected or damaged is going to be walled off by fibroblasts. So you'll make a wall around that area so the microbes can't spread around the body. And just to kind of show you with images what's going on with that. So here we have tissue damage. So you have a pin that's been stuck through the stratified squamous layer into the dermis. So we have chemical changes associated with that damage. Specifically, if there's microbes that have been introduced, we'll have mast cell activation. This is going to release histamine and heparin, and those are going to cause vasodilation to occur and increased permeability of the blood vessels. So here you have dilation of the blood vessels, increased permeability. Because there's blood flowing there, you may have various factors that will help to form a clot. Perhaps the blood vessel will even leak platelets and things to form a clot to stop the microbes from entering in. This will cause redness, swelling, heat, and also pain, all the signs and symptoms of inflammation. Not only that, we'll have Daya PD SIS, we'll have the white blood cells that will move out of the blood vessels. These white blood cells will undergo phagocytosis, and they'll also release cytokines, which will help activate other defense mechanisms. These phagocytes also remove debris by eating up those organisms and stimulate fibroblasts to wall off and to help to form a scar or scar tissue. We then will have the removal of all those organisms. We'll have clot erosion and removal. Scar tissue will form because of the stimulation of those fibroblasts. So pretty complex events that occur with inflammation and subsequent tissue repair. So something that occurs when you have infection in the body is the formation of pus. Pus are the dead bodies of microorganisms and dead white blood cells that have formed because of destruction. So all that dead material is what we call pus. If pus cannot exit through an exit wound, it will form an abscess deep in. I just showed you in this particular graphic an abscess in the tooth. So the doctor has to open that up and remove that abscess, or it'll be a problem area. You might have microorganisms still growing there. It's going to be painful, swollen, and has to be removed. So a boil on your skin is a skin abscess. So if you ever had a boil, a lot of times they'll lance those things, allow the infection to leak out, the pus and dead cells to leak out, and that speeds up the healing process. So lastly, we have fever. Fever is part of the innate response. Abnormally high body temperatures are going to be greater than 99 degrees Fahrenheit or 37.2 degrees Celsius. And this higher body temperature is induced by chemicals we produce called pyrogens. These are secreted by various leukocytes or white blood cells and also macrophages. So when they sense you have certain kinds of infections, they'll release these pyrogens and your body will begin to generate heat. A warmer body temperature is not such a bad thing, although it's going to make you feel bad because it'll make you have the shakes, shivering, you'll feel cold, and generally makes you feel kind of bad. Having a hotter body temperature, though, is probably really good for you. It makes your liver and spleen sequester or take out of circulation iron and zinc. These are needed by microbes like bacteria in order to undergo binary fission or their form of reproduction. So we inhibit microbial growth by sequestering these particular molecules out of our bloodstream. Fever increases cells to divide and to repair. It also causes a higher body temperature also causes greater vigor in phagocytic cells attacking microorganisms. Okay, so fever's not such a bad thing. I know we're quick to take an Advil or a Tylenol to stop fever or reduce fever, but that's it's good to have a little low-grade fever in order to fight off infection. So I think this is a good stopping place. In our next lecture, we'll talk about acquired immunity, and so we'll talk about producing antibodies and cell-mediated immunity, and talk a little bit about vaccinations. So until then, I will see you next time.